Good day, everyone, and welcome. I'm Jack Van Horn from the University of Virginia, and I'm very proud to welcome you to this edition of our 2022-2023 Foundations of Biomedical Data Science Seminar Series. This lecture series is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since 2016, the Foundations of Biomedical Data Science Seminar Series has focused on the use of data science methodologies, artificial intelligence, deep learning, and statistical modeling as they pertain to different themes in the biomedical and health sciences. This year's theme is data science and the public health consequences of the COVID-19 pandemic, where we will enjoy presentations on Fridays at noon Eastern from leading thinkers about the issues, promise, opportunities, and hurdles associated with understanding the secondary health effects of the COVID-19 pandemic. These include delayed health checkups and treatments, mental health concerns, drug abuse, use and abuse, increased obesity, and the long lingering health effects associated with the SARS-CoV-19 virus. Indeed, our 2022-2023 application cycle is now open, and we encourage junior faculty members working in areas such as infectious diseases, epidemiology, clinical medicine, population health, as well as data science, machine learning, AI, statistics, and other relevant domains to consider applying. We believe this is an exciting and engaging program where we characterize the secondary health effects of COVID-19 in a unique and creative way, quantifying and qualitatively identifying approaches that are new and lead to multidisciplinary collaborations. Participants selected for our Biomedical Data Science Innovation Lab program will leverage these presentations as vital material for a culminating in-person grant project development workshop to be held at the Lodge at St. Edward's Park in beautiful Seattle, Washington in June of 2023. Today, I'm delighted to welcome our speaker, Dr. Herbert Sauro from the University of Washington. He is a 2023 BDSIL mentor, and Herbert is a professor in bioengineering at the University of Washington in Seattle and serves as the director of the NIH Center on Biomedical Model Reproducibility. Originally educated in the UK, he wrote one of the first biomedical simulators for the PC way back in the 1980s and made significant contributions to the early development of metabolic control analysis. In the 1990s, he started two successful companies to develop simulation software for education and to provide consultancy to the finance industry in the UK, and has received many awards for excellence in teaching and a special commendation from the DARPA Information Processing Technology Office. Herbert's current interests include encouraging best practices in biomedical modeling, investigating new ways to create models that are dependable enough to be used in clinical settings. In continuing with our 2023-20 Biomedical Data Science Seminar Series theme, Herbert's lecture today is entitled Publishing Reproducible and Credible Models. Publishing repeatable and reproducible computational models is a vital step for the scientific method in computational biology, and one that is often forgotten in the rush to publish. The pressures of academic life and the lack of any reward system at institutions, granting agencies, and journals means that publishing reproducible science is either non-existent or at best presented in a form that's an incomplete description. So Herbert is going to talk today about both the formal and informal techniques one can use to ensure that our published work is reproducible. As always, we are streaming this lecture live and for recording via YouTube. And if you are watching on YouTube today, thank you so much for joining us. Also, our specially selected 2022-2023 Biomedical Data Science Innovation Lab participants and alumni are encouraged to submit any questions they have via the chat feature in their Zoom sessions. I'll synthesize all of these and I'll ask them with Herbert on your behalf in the last 10 minutes or so of the lecture. And with that, Herbert, welcome. Thank you so much for doing this. And we are really looking forward to hearing from you.
Thanks very much. I'm always happy to talk about the work we do. So I got a bit of a cold at the moment, so excuse me if my voice is a bit weird, but it's the what happens when you live in Seattle in the winter. So the first thing I want to do, actually, rather than, we mostly put acknowledgements at the end, but I want to put them in the front. So the work I'm going to describe is, I work within a community, and I'm a big fan of communities, and a lot of people are involved in the work that I'm going to describe. And it's interesting that a lot of the work has been driven by younger scientists. And it's younger scientists who see the advantages of promoting open science. And science is often a generational thing. It takes a generation for things to change. And hopefully, the new generation will come up and push the idea that science should be open and reproducible and, of course, credible. Okay, so let me start off with a completely different topic. This is one of my pet topics. Euclid's Elements. Some of you may have heard of that. It's a foundational book on plane geometry. It's 2,300 years of reproducibility. So these are some of I'm a bit of a collector. These are some of the copies I have. So there's a Greek edition, 1571, and from Strasbourg, and then Isaac Barrow in Latin, 1678, and then a new one from Dana Densmore here in the United States in English in 2013. The point I want to make is that I can reproduce Euclid's proofs even today after 2,300 years. If you try to do that with work published last month, you will find, at least in the systems biology community, that you can only reproduce half of them. And that's a good number. In most other fields, the reproducibility level is probably close to 0. So out of interest, I show you the picture of the two. The one on the top is the Barrow. They're really cute little books they used to make in this 16th century. They're sort of pocket size, but anyway. Just to show you, they can go back even further than 1571. This is the Stephanos Greek 888 AD copy. This is actually the oldest extant copy of Euclid. It was written in Constantinople, and it survived the ups and downs of civilization. I mean, I think a couple of civilizations rose and fell while this book existed. So it's always a big surprise when I hear that, material that was published last month has only survived about a day. But anyway, so that's my little soapbox anyway. So what I want to talk about today, is the issue of reproducibility in modern science. I mean, it's been talked about for quite some time now. These are two oldish articles now. One on the left was from The Economist, so it's appeared in the popular literature as well as in more formal places like Nature and so on. And this stemmed from a study that somebody did. They tried to reproduce some cancer-related wet lab work and found they could only reproduce a fraction of them, and that upset a lot of people, obviously. And then it went viral, as they say, and everybody started talking about reproducibility. And it's interesting, during the COVID, last couple of years with COVID, the idea of problems with reproducibility and then credibility arose in the COVID modeling area, because policymakers use computational models to decide on policy, and that policy affects millions of people, and it could, and we're talking about life and death situations here, and it could result in whether, a large number of people die or a large number of people survive. And there are two primary models that we used. The one on the left is the British model from Imperial College. That was highly criticized back in 2020, both in the popular media, but in the newspapers and in the more formal scientific journals. There was significant criticism of the way the software was written, and that it seemed to produce different results every time you ran it, which wasn't apparently due to any stochastic nature of the model. It was a rather strange problem. And then the other one was on the right. This is from my own university. This was used by I should say that the British model was used by the UK government, and the University of Washington one was used by the US government. And they both had their issues. I would say, though, that, as often the case, the issue was overblown a bit, because actually in hindsight, the model did a reasonable job, actually, of predicting infections and deaths, surprisingly, even if maybe, the software was a bit suspect, but it still actually worked. So, and there are various papers you can look up. They go through what they found and so on. So it's an interesting exercise, especially in computational models that are of national importance, right? Where governments rely on them, and big decisions. I mean, billions and billions of dollars are moved around depending on the predictions from these models. So reproducibility has a life and death aspect to it, but of course, in our own academic work, we don't deal at that level, but we still should be doing, proper science. So our work should be reproducible, even if it doesn't affect people's lives so much. So one other thing that I want to say, even with all, all the newspapers, and journals saying there are problems here, slightly overblown, but there was some study done, well, actually it's over a year ago now, came out as a preprint, where they looked at all the papers in the various preprint servers with respect to whether the papers gave you access to the data and models. So there's a preprint here that studied a lot. I mean, they studied thousands. They went through thousands of papers and did a study. And so they're looking for basically either open data or open code. So they looked at, you can see they looked at 4 preprint services, the original archive one, the bioarchive, medRxiv, and soc archive. As you can see, a huge fraction of them did not give you access to the data or code. And the histograms on the left are over time. They show you the proportion of papers that had code or didn't have code. So the red bars are bad. Okay, so a lot of papers just didn't provide all the necessary information. So we're still doing this kind of thing even today, and it's a major cultural problem actually, that we don't provide all the information necessary to reproduce a given study. Now, my focus, of course, my particular area is in systems biology, and the reason why the COVID thing came up was topical at the time anyway. But also, it turns out some of the technology that we developed in the systems biology area are now being used to encode COVID models, which is interesting. So there was there's been some overlap between the fields. But my area is systems biology, and we've looked at for many years actually, before even everybody got. Before reproducibility hit the newsstands, we've been looking at how reproducible models were for at least 15 years. Because once we developed the standard, the SBML standard, which is a way of exchanging models, which I'll talk about more later, suddenly there arose the ability to have repositories of models. And then once you have repositories of models, you can start doing metadata studies on those repositories. And I know, like from 10, 15 years ago, that there were, as the repositories were putting models onto the databases, they were having to curate them. And so they were collecting statistics on how often they could actually reproduce a study just from the paper. And there were 2 repositories. One was Biomodels, then was PMR down in New Zealand. Biomodels said that 97% of all models couldn't be reproduced, and the New Zealand group said that 100% couldn't be reproduced. Now, this is anecdotal in the sense that it was never published, but last year, actually now, time flies, 2021, the Biomodels group in Cambridge actually published a formal study, and you have to think of this as 10 years later now, and it's now 50% of published models are not reproducible. So there's been a huge improvement in the reproducibility of models, and this is primarily down to the use of standard exchange formats. Okay, so you have a formal method for representing your models and distributing them in your papers. So this is good news. It doesn't look good news, but it actually is good news because it's a significant improvement. So you might be interested in, well, why aren't these papers reproducible? What are the issues? I mean, they're all very mundane, actually. And I've got a selection here. There are hundreds and hundreds of these, okay? I just got some here. For example, the first one on the top left is a HIV/AIDS one. There weren't enough. They didn't describe all the parameters. The second one is the model was incomplete. You couldn't actually even run the model. Another HIV one, there were no parameter values. The NF-kappa B one is a very interesting one on the top right. That's a very famous paper in Science. It was one of the first papers to show oscillations in the NF-kappa B signaling network. And it turns out you can't reproduce it at all. So it's irreproducible. Then on the bottom, which is a funny one, on the bottom left, lessons learned from quantitative dynamical modeling in systems biology. The analysis procedure wasn't described, which I thought was an irony given the title of the paper. Another one, parameters incorrectly annotated. I'll come back to that one in a minute because I'll show you exactly what happened there. Next one, irreproducible. And then the third one, this is a famous paper in Cell by Jonathan Carr, a colleague of mine. They did one of the first large whole cell models, right? You're modeling the entire cell. This is a mycoplasma model. And the whole thing was written in MATLAB, and it meant that it was a large amount of code. And it meant that you really couldn't figure out what was going on. And I'll have more to say about that in a minute. Let me go back to the this parameters incorrectly annotated. Oh, okay, let me just summarize this first. So these are the kinds of problems you encounter. Very mundane, right? Missing data, incorrect data, results not are published without models. That happens even now. There was a couple last year that did that. The models are only supplied as binary, which is really useless. The simulation environment is not available, and this happens more and more, of course, as the paper gets older and older. The wrong model supplied with the paper, and then this one, not so often now, but back in the 2000s, because people loved to have their own website, and they would put their stuff on their own website, and then eventually their website would go away along with all their data and so on. And so a lot of the old papers, you can't get the model anyway because it was on their personal website, which has effectively disappeared. So let's go back to that other paper. It was 2011. This was one that a colleague from industry actually sent me. He was very frustrated. He said it took him a week to get this model working. It was a neurophysiological model. I mean, a week is pretty short, really, but I mean, he was probably, who knows how much he was paid, and that money was basically wasted in trying to get this model to work again, which they did eventually. And just to look at this more closely to see what actually went wrong, there's a couple of things to note here, some practice issues. One is, if you look on the right-hand column, and the second paragraph and the first paragraph, if you look closely, you'll actually see the model embedded in the text. So these are differential equations. So you look dn by dt and so on. So that we don't recommend that you actually put the model in the text itself. But you know, even if they did, right, my colleague of mine from industry, he couldn't get this working, and he discovered the problem was that the units. So this is a neuron. Okay, so a neuron is basically a cylinder. It has a diameter and a radius, and that 23 micrometers is actually the diameter, not the radius. And so if you assume it's the radius, of course, the volume changes completely, and that affects all the reaction dynamics. And of course, that's why it didn't work. Now, the other thing was there was an additional area that wasn't included, which was the ends of the cylinder, which they forgot to include. So anyway, it's like hunting for a needle in a haystack, right? Looking for where the problem is. And it sometimes can take months, depending how big the model is, take months to get a model working. I mean, some could argue, well, that exercise is good anyway, especially for a graduate student to get to learn modeling. Well, yes and no, but there's a lot of effort wasted in trying to get published work working again. Now, you know, I talked about reproducibility, you know, as an important issue, but there are other broader aspects that we should think about as well. So when it comes to reproducibility, we're basically saying, can I recreate what you did? Okay, that's an essential aspect of the scientific method, right? If you can't do that, you're essentially not doing science. Transparency, I mean, can we actually even understand what you did? Or maybe I can, you know, get the same results, but is there any way I can figure out, you know, what you did, and can I understand it? And that'll get me back to the large scale mycoplasma model. And then the other thing, which is really becoming more and more important, and that comes back to the COVID stuff I said at the beginning, whether the model is sufficiently valid enough that you could actually use it in a real world situation, you know, trust it. And this gets back to a more broader idea of credibility, right? How credible is your model? Is there a way to measure how credible your model is? And I'll talk a little bit about that towards the end. And that's something we're really interested in at the moment, is how can you assess whether a given model is credible? And there are all sorts of metrics you can think of to measure the credibility of a model. And then this goes back to the acronym FAIR, which some of you may be familiar with. This came out of the UK, I believe. It's now promoted by the NIH. This is findable, accessible, interoperable, and reusable. All right, these are the things we would like your data to have, the properties we'd like you to have. And unfortunately, most published research is not under the FAIR umbrella. There's usually one or more of these things that are not there. Okay, but then the question is, does it really matter? Okay, how much does it really matter? I mean, you could argue, a lot of studies come and go, and they're rarely cited. And how important is it that we put the effort into making sure that work is recitable? I mean, I think it should be obvious. I hope it is obvious. I got a cloud up there saying, I hope you think that this is obvious, but that for nationally important issues, right, such as pandemics, climate change, economics, etc., those models should be reproducible and credible, of course, since policy decisions are often made based on models today, right? And with AI and machine learning becoming more and more, prevalent, that's going to become even more important, because AI models, we have no idea what they're doing. There's zero transparency, and it's even more critical that those models are tested thoroughly. But what are other concerns, that are maybe not of national importance, but that concern us? Well, first of all, there's a loss of intellectual capital, which is a waste of tax funds. And we always complain about writing grants and there's not enough money and so on. Well, maybe we shouldn't be writing stuff that can't be reproduced and takes people effort then to reproduce it, and that costs actual tax dollars. There's also the element of professionalism. We are a professional body. I mean, we're paid a salary, so there's some element of lack of professionalism. There's also it's a poor application of the scientific method. I mean, the scientific method relies on being able to reproduce another person's work, because we do that in order to gain confidence that a given result is likely to be correct, right? But for me, actually, probably the worst thing that could happen, and is happening to some degree, is the loss of trust by the general public. We see that all the time now, from climate change, evolution, and of course the COVID thing was, there was a lot of mistrust by the general population. And the minute you lose the trust of the general public, I think you're lost. It's very hard to get back, probably. So, even if you don't care about the other three, the loss of trust by the general public will be devastating to our society. So, for that reason alone, we should be concerned about that our work is reproducible to everybody. So the question then is, okay, it does happen, unfortunately. The question is, why does it happen? Well, it's pretty easy, really. Our incentive system is skewed towards less important outputs. That's basically what's happening. Because, science has become a professional, well, I guess it has been a professional enterprise for quite some time, but these days even more so, especially with the government pumping in funds into science, that the incentive system has become skewed. And in particular, a lot of the universities and review panels, I've been on these, you see it. We tend to reward quantity over quality, because you can count quality, right? If you're a bean counter, you can count quality. Quantity, but quality is a much harder thing to assess. It requires expertise. And so, review panels, and especially promotion panels in universities, then tend to gravitate towards quantity measurements, like how many papers did they publish, in what journals did they publish, how many grants did they get, etc. Now, these pressures are very, very strong. It's hard to avoid them. I mean, we have limited bandwidth, and as much as we'd love perhaps to do the right thing, we inevitably gravitate towards these rewards, because promotion means access maybe to better labs, certainly means improved salaries and so on, which if you've got a family, that's important. So now Yeah, so there are some issues then to worry about here. As an anecdote, I remember hearing years and years ago that members of the civil service, for example, one time rewarded with more carpeting in their offices. So as they were promoted, they got better, bigger carpeting. Maybe we should do that. Maybe that would be better. I don't know. But a lot of these things that we give our colleagues, like better salaries, promotions, awards, better office space, don't always necessarily lead to better science. But you say, what about the journals? Surely they are the ultimate and final gatekeepers and will make sure all the work is reproducible. Yeah, you might think that, but unfortunately that doesn't work either. There's a lack of reviewer rigor at the publication stage. I see that all the time. There's also, unfortunately, a lack of interest by many journals, maybe I say most, in publishing reproducible work, but not all. And why is that? There's a couple of reasons. Well, I guess the main reason today, I guess the reason has always been there, but I call this there's a pay-to-play model, which otherwise known as open access model. You can probably tell I'm not a big fan of the open access model for that reason, because it creates considerable inequity. But anyway, it's a lucrative business, and journals are reluctant as a result to put up any barriers that make authors pick an alternative journal, right? And as a result, and I've heard this from journals, no one wants to be the first to require studies to be reproducible. Either all the journals do it at once or not at all, because when the first journal does it, they will immediately probably lose authors, right? Submissions, excuse me, because of the barrier they put in place. Now, there's an interesting story, though. The Journal of American Political Science, I believe it is, they actually did this on their own. They started to insist that all papers had to go through a third party to check whether the work was reproducible or not. This journal published a lot of econometric papers, so a lot of statistical analysis and so on. And they found that the prestige of the journal rose, in fact, because now people would trust it. And I'll have something else to say in a minute about this other aspect, that it turns out that people were then more likely to cite it because it was more trustable. And so the prestige of the journal went up. So, I mean, maybe they shouldn't be so shy in doing this, because they might find their prestige going up, and because journals are always looking for ways to increase their prestige. So, well, are there any solutions? Unfortunately, the university reward system is unlikely to change. It would require a major cultural shift. There's too much money at stake, and it's probably unlikely to happen. I mean, over the last 5 years, we've gone through all sorts of thoughts and discussions on how we can encourage reproducibility. At the end of the day, we think probably the most effective way is at the reviewer stage. If we can persuade reviewers to be more vigilant, and there's a simple we can do some simple things just at the beginning, right, to help matters. One is, does the paper clearly point to the source code used in the study? Does the paper provide the data required for the analysis, preferably the raw data? And then, really important, which is why I got in red, do the authors provide documentation on how to run the code and regenerate the simulation results? I mean, you can sometimes find the code, right? They just drop it in GitHub or somewhere, and you've no idea how to run the thing. So then you have to end up reverse engineering the code to figure out how to run it. And so providing documentation is actually crucial. So these are simple things as reviewers we can do. And you can tell our editors, associate editors, I really think you should not publish this paper because this and this is missing. And it's an easy thing for the authors to fix. And my experience has been, excuse me for a minute. Excuse me, sorry, got a cold. My experience has been, if you do that, authors are very quick to fix it, especially if the paper has been, accepted on condition that you supply, XYZ. Authors are very, very quick to do the fix. So, I mean, this tells me that this is not something that authors are doing on purpose. It's just, the last, you just want to get the paper out. You've got other things to do. You've got grants to write, et cetera, et cetera. You just push the paper out, and the reproducibility bit sort of is sidelined. That's what happens. It's not that there's any evil intent to hide things, although it can happen actually. So authors are very, very keen to do it if they're told that something is missing. Can journals be more active? Yeah, I mean they can, but they're not really. I mean, as I said, the reviewers, we need to be more vigilant, and I'm speaking here to the young scientists in the audience. You're going to be the ones that are going to be leading the scientific community in the years to come. You can make the change. There's a lot of resistance in the, as I said, in the Systems Biology journal, which is what we've been focusing on. 4 of them, though, are actually interested. And in fact, PLOSCOMP Bio, we did a pretty big pilot study with them a year or 2 ago, and there was something published on that. So they're pretty keen, but they're still a little bit hesitant because the minute you put up barriers, people, authors will start thinking of going elsewhere. But they're aware of it. They'd like to do it, and then the other 3 are also quite interested as well. But there are many journals that are either indifferent or actively against it. So, which is unfortunate. All right, so let's switch to a slightly related topic here. This one of terminology. There's actually a lot of confusion in the literature about what we mean by reproducible anyway. I got this little funny thing from Game of Thrones. It's a bit like that. There's a couple of papers here I cite, one in Frontiers in Neuroinformatics, a brief history of a confused terminology. But a great paper is actually a preprint by Barber from George Washington University. So a really nice overview of the different terminology that's used in different domains. And she comes up with this basically three large groupings that use the terminology in a particular way when it comes to reproducibility. So, and it can be quite confusing. And we basically focus on just 2 words. They represent 2 extremes. One is repeatability and one is reproducibility. So repeatability is where you've done a scientific experiment. Next day you do it again, and the same conditions, you get the same result, right? You basically repeated the work, you got the same answer, and you're happy. Maybe you'll do it a 3rd time as well. Maybe that's basically repeatability. That's what most computational science is actually in the end. So somebody will publish their MATLAB, Python, whatever code. You grab it, you run it on your machine. Oh, look, I get the same answer. All right. It's not a very strong statement because you're also repeating any bugs that were in the original code as well. So repeatability is really for the original author to make sure that the results they're getting are consistent. The other the other extreme, I guess, is reproducibility, which is where now you read the paper and you actually, from scratch, you redo one or more elements of the study. That could mean you collect the data again yourself, you reanalyze the data yourself, you rewrite the software, you compile the existing code on a different compiler, or you use a completely different operating system, or you use a different algorithm, but similar to the original algorithm. All these things, right? Stress testing the original study to make sure that the results the original authors got is actually reproducible. So that's what we mean by reproducibility, and we keep it to just 2 words. I mean, some people have all these different words to represent all different kinds of reproducibility, but we just stick with these 2, repeatability and reproducibility, to keep things simple. Now, an example of repeatability, a repeatability study is by a colleague of mine, Titus Brown, from Michigan State University in 2012. He's getting old now, I guess, but he was one of the first to show, just in a genomics domain, where people do umpteen analyses on the data, lots of statistical work and so on, and massaging I wouldn't say massaging, but cleaning the data, making sure the data is okay. Inherently very difficult to reproduce because there are so many stages in the process. So Titus Brown and his colleagues, they published this little preprint on an example of what you could do if you wanted to publish a paper like that. And they basically containerized the entire thing in a, I don't know if it was a Docker. I'm not sure if Docker was around then, but it was a container where everything that you needed to run the study was in this container, and you could just run this container, especially a virtual machine. You could run this anywhere then you wanted, and you'd get exactly the same result. So this is an example of a repeatable study that was done, which is quite nice. So he gives a couple of things here, like they had a tutorial, they had a Git repository. Nowadays, you'd put it on GitHub, I guess. Instructions, an IPython notebook, right? So they were using these sort of notebooks back then before they became popular. He has a pipeline to run the data processing, as well as the assembly. All right, so that was an example. Now, you might think some of you may be familiar with workflows. You may be familiar with some of these workflow engines like Galaxy, Taverna, Pegasus, and so on. They were originally designed in the hope it would solve this problem of describing a complex analysis of data so that you got to hand over to someone else and they could just do the same thing. To some extent it worked, but to another extent it hasn't worked very well at all. Couple of problems. One is a lot of these engines, they use their own proprietary formats. And so you're stuck with that engine. If that engine dies for some reason, like, it's no longer funded and the source code is not, and, the engine is no longer maintained, you can't run your workflow anymore. And you can't move it to another more modern workflow engine because they're using their own particular language. Because each of these three use a different way of describing a workflow. So workflow is just a way of saying, you start with some data, or you're starting with a database, and it tells you, pull this stuff from the database, do this analysis, then do this, then do this, then do this. And each time is telling you which application you should run in order to process the data. It turns out to be very fragile as well, and fragile in the sense that you may be given a workflow, but one of the applications it used is either no longer available or it's been upgraded to such an extent that it can no longer be run within that workflow. And this happens a lot. There's a workflow sort of repository at myexperiments.org. Many of them there can no longer be run because there are dependency errors, or the workflow engine is no longer available or compatible or whatever. And in fact, there's even a term that describes this. It's called workflow decay. So although many people may advocate workflows for reproducibility work, I don't recommend it. I mean, it's a tough one. You'd have to make sure you include everything in some kind of containerized platform. Okay, so the other thing is, okay, we don't have a workflow, but we just have code. I could just distribute the code, surely, I could, distribute my model and whatever I've used, MATLAB, Python, C++, whatever. You could. It sort of works for small models, but it most definitely doesn't work for big models. So this is the Mycoplasma model I mentioned before. By Jonathan Carr, it's about 150,000 lines of MATLAB code and 2,000 lines of Perl. The data is stored in binary files. So there's a workshop in 2015 that tried to reproduce the model, but was unsuccessful. It was very hard to try to recreate the model in another language, mainly because as you read the MATLAB, you're relying on comments to tell you what each step does. And it's sometimes the comments were out of sync with the code, like the code had been modified, but the comments hadn't been updated. So it turned out to be incredibly difficult to reproduce. So the lesson here was this is not the way to do it. And fortunately, people are still doing it this way. For large models, it's fatal. It's basically unusable because it's just one large monolithic lump of code. Yeah, I mean, the problem was hard to understand how they built, what assumptions of data were used. Very hard to reuse in other projects. And the executable environment they originally used may no longer exist, and then you're just really stuck. So summary, just to what can we do? I mean, the simple things we can do, and these are really simple things we can all do, is when reviewing papers, we can ensure that everything is at least available. So in principle, it could be run and should be in a prominent place, not, for example, a figure caption, which can be easily missed. And also check that the authors have provided the instructions. And then for the more sophisticated, you could tell them, store your stuff on things like GitHub, or better still, Zenodo, and obtain a DOI. And then provide tests. Basically, in the computing industry, they're called, I've forgotten the phrase now. Unit tests with your code to show that your code can be trusted. Okay, and you can imagine some sort of, this is just a fake thing I put up, but you can imagine some sort of scoring system that a journal could do, depending on what the authors have done. All right. Okay, let's go on to a new topic now, and the way we have tackled this problem in systems biology. One way is to galvanize the young, because they are very enthusiastic. And this is basically what we did in the systems biology field. So in the systems biology field, the kinds of models we're talking about are mechanistic models, all right? Metabolic pathways, signaling pathways, not AI models, not statistical models, okay? Those are the kinds of things we're talking about. So a little bit of history, around 98, 99, several of us were frustrated that we couldn't exchange models between different modeling tools because the formats were incompatible. This was particularly a problem because there's a very high turnover of academic software, very short lifespan. And you can't really rely, if your model is, if you use a particular application and, 5 years later, the application no longer exists for some reason, you're stuck because you probably saved it in their format. And now somehow you have to convert that format to a new tool if you can. And you're basically left with orphan models, which is a big problem. So we wanted to get around that problem as well. In fact, all the tools back in 98, 99 that we used no longer exist. All right. The source code isn't even available online anywhere. So that was our impetus. Okay. And then in parallel, luckily, there was a major uptick in interest in systems biology. I mean, we were a small little group then, systems biology hadn't really, the phrase wasn't really used back then. And then suddenly in around about 2000, everybody got interested in it. And there was a lot of funding reappeared. In particular, the Japanese Arato program wanted to fund some infrastructure in systems biology. And ourselves and the Japanese group got together, and that resulted in this thing called the Systems Biology Markup Language, which was released in 2001. And this basically solved the exchange problem. So, and to my surprise, it really spread really fast. And now pretty much all tools in systems biology support this language. So I can take a model from any tool, save it in that tool, and load it into another tool. And it doesn't matter if that tool dies one day and disappears. I've got the SBML, and I can just load it in the latest and greatest application. And the other thing that happened was, to my surprise, was it spawned a whole new community focused on all sorts of other things related to modeling, such as diagrams. How can we represent diagrams in an unambiguous way? Oh, now we've got a standard. We can have model repositories. We can start annotating the models with new ontologies. And that led on to another standard called SBML. Well, now we could describe how somebody did the simulation experiment. So I'm not relying on a program script, but I have a description of what was done, which can be converted into a programming script. And so out of this then formed this organization called Combine, which is a really active one. We have 2 meetings a year. Very grassroots effort. That's the emphasis, that this stuff is very grassroots, bottom-up effort, not a top-down. Okay. All right, I'm going to move on here so we can finish. What's the point? This is basically what I've said, having modeling standards. We can exchange between different tools, makes models much more likely to be reproducible. they can be now understandable because you can annotate all these models with the information, like what assumptions I use, where did I get the information from, and so on. And it's all computer readable, and it's now searchable, et cetera, et cetera. And it makes it much easier to reuse the model. Okay, you can now reuse them. Interestingly, there's a study, it's under review, I know, from Germany, and this is a nice big fat carrot for all those researchers. It turns out that if you use, in systems biology at least, if you use these standards, your papers get more cited, more citations. So if there wasn't a greater carrot than that, I don't know. And I basically boils down to if it's in a standard format, it's easier to reuse, and therefore it's more likely to be used, and therefore the original paper is more likely to be cited. Whereas those papers, it's really hard to get the model out. There's less of a chance of it being recited. So, and part of our center, we wrapped all this up into a major new portal called Biosimulations. The infrastructure is in place. We're now filling it up with models. Basically, this is a place where you can go, search for a model, search for an article, and then it'll give you the model, and then you can run it there and then to show that you get the same results. And then at that point, you could download the model and run it in your own favorite application. And we support a huge number of standards, a huge number of modeling approaches. So we're very excited about this. So hopefully this year we're curating a whole bunch of models on biomodels, working with the biomodels team. In fact, last year, we completely recurated the entire biomodels database for them to make sure everything was properly reproducible. Just then briefly, then what next? Because this is a work we're trying to get funded. Now that your model is reproducible, okay, what's next? Well, what's next, to me anyway, is model credibility. And I'm part of the IMAG, or the MSM, the multi-scale modeling community, meets every year in Bethesda. This is a multi-agency organization. They've started thinking about this. So Jerry Myers is one of the authors here is from NASA. And after the various accidents they had with the space shuttles, they looked deeply into the idea of making sure that the models they were using were credible, so they didn't have the same mistakes they had before. And then he teamed up with a bunch of biomedical researchers and came up with a sort of 10 simple rules for credible practice. And so this is sort of the start then of an effort now, and I was briefly part of this as well, but we'd like to take this much, much further. At the moment, this is a list of things that you can manually check to rate whether is the paper using credible practice or not. But what we want to do is go much further. Now that we've got all our models stored in computer readable formats, we can do deep dives into the model to find out whether there are any simple mistakes. Let me go to this slide first. So we want to split the credibility tests into static tests and dynamic tests. So static tests are things like, are there mistakes in the naming? Did they use NAD caps or NAD lowercase in one place and not another? In a very large model, such errors are very difficult to spot, right? So we want to create a whole toolbox that lets people check these sort of mundane problems, but can be easily spotted and fixed. Any problems with the rate laws? There's a whole nonsensical processes, right? Glucose going to sulfur dioxide. That's a bit of an extreme example, but any uninitialized stuff that can be very difficult to spot if you're not careful. Checking for mass balance, all sorts of checks that we can come up with. These are static tests. The model's not run. But then once you've gone through the static test, you can start doing dynamic tests and say, well, what are the kind of numeric tests are there? Are things going to infinity, going negative, doing some strange stuff? Can we validate? Is there any validation data that can be used? Can we verify the model in the sense, can we run it on a completely different platform and get the same result? And the verification thing is something we can now do on our biosimulations platform, because you can just ask the platform, we can run this on a completely different simulator, right? And cross your fingers, you get the same answer. And that's a nice verification test. So we'd like to know in the next phase, build up something to do related to model credibility. And so that's basically where we are. So I think I'm dead on time. That's amazing. 10 seconds to go. So that's it for now. And I'm looking forward to any questions you may have. Thanks very much.
Herbert, thank you so much. As you were kind of talking, I was just kind of wondering, you sort of touched on it a little bit.
Yeah.
What do you think the role of peer review is in this process, a little more deeply? Like I'm a journal editor, I chase reviewers around.
Yeah.
Most of the time they decline a review invitation.
Yeah.
And yet, very often it's those same people who are complaining about the state of the literature, right? And you're like, you had your chance.
Yeah.
What from your
Yeah.
perspective, what's the
Yeah.
role there?
I
'Cause it's
hear
so
you.
important.
I hear you what you're saying, actually. I know that's why I don't want to be an editor. I mean, I was once, and it's a tough job. I think the universities have to step in and say, this is part of your service, and formalize it a bit more. And that may you might then find it easier to find reviewers. I know it's sort of the university micromanaging us, but if as a profession we are not doing our professional job, then somebody higher up may have to step in and say, look, by the way, you really should be doing this. And the societies too. I mean, that's why we have societies, right? The professional societies, they meant to encourage best practices and good citizenship amongst all the scientists. And this is one thing they could do as well. So two things, the societies and the universities. And the nice thing about asking the universities to make it a service part of the work, it doesn't involve money, which you could, like grants and things, which the universities don't want to touch at all. And it's an easy thing to do, right? You can say in your promotion packet, they can say, well, how many papers did you review? I mean, people can game it, of course, just by doing cursory reviews and not doing it properly. But then the editors can come back and say, or, the website can say, did you check the following? I mean, that's another way of doing it. But the journals are very reluctant to do anything like that.
Yeah, it's a challenge. And
yeah.
It's a challenge just to get people to review. And then the reviews that you get are often not a deep dive. And certainly, even if the stuff is available, the code, the data is available, people aren't necessarily going to go to the trouble to actually go and run them all through. So anyway, I'm just kind of curious because especially with things like the various pre-publication archives, where peer review doesn't happen, right? Now you've got a internet full of these repositories, which is on the one hand a good thing. On the other hand, it's non-peer reviewed stuff. So no one's passed any sort of credibility test on it. I
mean, here's a thought. I mean, with AI being so sophisticated these days, maybe we could get a chat review app that just goes through and checks for us. And just writes
maybe it'll just write the paper for me too. And
well, it'll do that already now. But it could automatically write a report for you. It could be your third reviewer. In fact, we offer actually as a service, the center, to be a fourth reviewer. So if you've got 3 reviewers, we're the fourth one. And all we don't do a technical assessment. We just look at whether the data's there, the models are there, and there's enough information to at least try to run the model. And then we did that with PLOS Comp Bio. Worked quite well, I think. We did a pilot study with them, and it worked out quite well. And so we're hoping to pursue that again with them. Because it doesn't cost anything then. There's no change to the infrastructure. All they have to say is they just have to the editor just has to add a fourth reviewer, basically. And we will always accept the job, not like a normal reviewer. So
that's nice to know.
Yeah. And we take a week or two. We're pretty quick turnaround as well. We have a dedicated team that does that. And we could do the checks for you. But in long term, I think an AI solution might be the way forward, actually.
Well, another thing I was thinking of as you were speaking, and you started off with a really fantastic example of Euclid's Elements, and that it had survived the rise and fall of various civilizations. It'd been
Yes.
through multiple world wars, you know, and yet it has this enormous staying power. And the principles and, you know, concepts that are laid out in that are just universal, and it's part of math, right? It's just what we understand as foundational math. And yet, in contrast, the stuff like you were pointing out that gets, you know, created last week, you know, is already old, right? And I'm just kind of curious, what does it take now to actually have something that's like a, you know, Euclid or Newton or, you know, something else that's going to have, they're going to basically help to reinforce the bedrock of science, in contrast to derivative, small-minded, you know, rush to publish, small sample size kind of stuff.
I'm with you. I know. I'm so with you on that one. And
is data science going to be able to help give us some ideas?
I mean, we don't think deeply anymore. I don't think. I mean, some people do, of course, but the pressures of funding and short-termism means we don't have time to think too deeply about things, and everything is very incremental. And I mean, I really don't like the way things are done today like that. I mean, well, I grew up, you know, in a time, the early 80s, when I was a PhD student, a postdoc, and my advisors were from a time where that didn't happen. And one of my advisors in Edinburgh, he would take 2 days to review a paper. And that's all he did for 2 days.
Yeah.
And he would actually contact the authors, actually, if he had a problem. That never happens now, I don't think.
It used to be that there was I don't know, there was some honor in it, right? It was a noble thing to review a paper, right?
Yes.
And I remember a colleague of mine, even, you know, not that long ago, would sign his reviews because he felt that it was important that the authors knew who was evaluating the work, and he felt it was important to sign them. And that was kind of considered controversial. You know, I can, you know, but then you also think about things like, you know, the Royal Society, for example, and how, you know, giving, you know, demonstrations of the science in front of people instead of just taking your word for it in the printed page. Anyway, it's a very different time, and I'm just kind of curious about the ephemeral kind of, you know, digital technology that is, like you said, you know, things change and all of a sudden you can't use it anymore and it goes away.
Well, I should say, I mean, the SBML stuff that we did, I can still run models that were published almost 20 years ago, because they're in SBML. And NSF had this thing, I think, at one time, that your stuff should be available for at least 10 years after you publish. I don't know if they do that anymore, but if you if you separate the description from the execution, right, that this kind of thing will work, because now you have the description in a formal way. You can convert it to anything that comes up in the future. I mean, the latest thing is now Julia, right? So now there's software that can run models from 20 years ago in Julia. Whereas if
I mean, it's an interesting phenomenon too, though, about like the software, you know, of the month, right? At the moment, it's all Python. I've seen this movie before,
right?
You know, next, you know, I don't know, in the future, it'll be Julia, and we don't use Python anymore. And you're like, but I went all in on that.
Yes, right. So so for us, the separation of description from execution is really important to us, because that you do decouple it from dependencies on the flavor of the month. So,
I think that's probably very important to make sure that you're not platform or software model dependent, that things, you know, they transcend the computer language the code was written in. And I suppose that, you know, for Euclid, that's probably true. If Euclid would have written everything in Python, we would have forgotten it by now.
We'd never know about it.
Exactly. But he chose to write it in Greek, I guess.
Greek. Well, even that was a problem, because then you needed translators. And it ended up in the Arab world. It was translated to Arabic, and then it had to be translated to Latin, and then from Latin to English. So, you know.
Given that everyone is kind of overwhelmed with writing and doing and,
Yeah.
and reviewing and everything, is there room for some combination of an automated approach and a human in the loop kind of approach to review articles and science and free up time so that people can do more deep thinking?
Well,
Is there an AI assisted review?
Yeah, I think definitely. I mean, thinking about, at least for checking certain aspects of the paper, that an AI could be useful, technical aspects, not the scientific side, but the technical aspects, like have they provided sufficient supplementary information? I'm sure you could train an AI to do that.
I know the journal I am the editor for, we have plagiarism detection, which is very
there you
handy.
go. Oh, there you go. Yeah, it's basically you can extend that to checking, and that would relieve a lot of pressure. But in terms of short-termism, the funding mechanism has to change.
Yes.
We're too obsessed with incremental and also now more applied work. And I think we're losing out on fundamental work and the next big discoveries that will come about. I mean, CRISPR arose because somebody thought, oh, look at those repeats in E. coli. I wonder what that's for. I mean, would that be funded today? I don't know.
Yeah, it's again, the way in which the system is structured to think about how to fund research is often has a event horizon. Somebody was explaining the other day that, oh yeah, we get grants in 5-year intervals, but the NIH tends to think in about 3-year intervals, and then there's turnover within the NIH as well, and so things kind of get forgotten and everyone wants to create the new, and we forget about these things. At any rate, it's these are really interesting, and reproducibility, credibility are vital. And Herbert, I want to thank you so much for spending your time with us this morning. And
it's been fun.
Really
I got a chance to show my Euclid books.
Yeah, that's awesome. And thank you so much. And everyone, thank you for joining us. And we'll look forward to chatting with you, with everyone again next week. Thanks again, Herbert.
Bye now.